Hello. Today in this presentation, I will discuss our proposal to remove motion artifacts from the measurements of a wireless electrocardiogram. The talk is entitled as Application of Tensor Decomposition in Removing Motion Artifacts from the Measurements of a Wireless Cardiogram. This work is a joint research effort by one of my PhD students, Yanis Lilienthal, and myself. According to the World Health Organization report published in 2019, more than 70 million people died of cardiovascular diseases worldwide. About 80% of the deaths could have been prevented by using early detection and monitoring methods. The Center for Disease Control also published a report in 2019 supporting this observation. Cardiovascular diseases are the prevailing cause of death in the US. Consequently, there are different types of handheld and wireless devices available to monitor heart rate, to monitor cardiovascular activities, and provide people early warning as well as recommendations. One of the most frequently used devices to measure and diagnose cardiovascular activities is the electrocardiogram. The electrocardiogram monitors the heart's activities, the heart's electrical activities, and I will give you a brief overview how this can be achieved. The heart, as you might know, consists of 4 parts. The upper parts or the upper chambers are the left atrium and the right atrium. Jointly, they are called atria. And the lower part consists of 2 chambers, the left ventricle and the right ventricle. The atria momentarily collect blood coming from the body as well as from the lung. And then when these chambers are full, blood is poured into the lower chambers. The lower chambers then pump blood forcefully into the brain, into the lung, and into the rest of the body. This activity of the heart is controlled by the electrical conduction system, the cardiac electrical conduction system. And I will briefly explain how this functions. The heart produces periodic impulse, and the source of this impulse is the sinoatrial node, which is located here. It is also called the pacemaker. When this electric pulse is generated by the SA node, it travels through the walls of the atria. We consider only one of the atriums for our case. Let's say this is the left atrium. It takes some time for the electric impulse to reach the AV node here, the atrioventricular node, during which time the blood is accumulated in the atrium. When the electric arrives here, blood is already full, and then the electric system now leaves the AV node. During this time, the valves blocking the blood from going into the ventricle will open and blood will go into the ventricle. Now the electric impulse leaves the AV node, goes through the His bundle, this nerve fibers, collection of nerve fibers are called the His bundle. And then from the His bundle, it branches into the left bundle branch and the right bundle branch. And then from this bundle branch, it propagates to the Purkinje fibers. These thin fibers are fused into the ventricular muscles. And when the electric impulse reaches the Purkinje fiber, it tickles them. As a result, the ventricle is contracted, and this contraction forces blood to pump out of the heart. So the clean blood goes to the brain and to the rest of the body. The deoxygenated blood, which requires filtering, goes to the lung. To the lungs. Now, this is how a normal heart function. If any of these electrical systems or the walls of the heart or the blood vessels are affected, then it affects the electrical activity of the heart. And by closely examining the electrocardiogram signal, it is possible to localize the problem and search for some remedy. The electrocardiogram, you might be familiar with it, looks like the following. It has three main hills or wave and a silent period between these waves, and each has a meaning: the amplitude of the signal, the shape of the signal, and the distance between the waves has a very useful meaning, and doctors usually analyze this to determine what is wrong with the heart. So here we have the P wave. The P wave is associated with the activity of the atria. When the signal propagate in the atrial wall, this wave will be observed. And after the valves are open and blood pours into the ventricle, there is a silent period. And so this period, this segment, refers to the time the blood takes to fill up the ventricles. Once the ventricles are full, then this QRS wave complex will be observed. This is the result of the depolarization of the ventricles. And this is a very critical wave complex to observe so that we know the heart is properly functioning. Then there is some relaxation time, and then the ventricle repolarizes, and as a result of this polarization, we observe the T wave. And then afterwards, we also observe the U wave, but the U wave is usually difficult to observe unless we use a highly complex device. In a clinical setting, when the person is resting and relaxing, doctors usually use a 12-lead electrocardiogram to observe the heart's electrical activities. And these are some of the locations where the electrodes will be placed. The electrodes are simple conductors, and they measure the potential difference between these points. The potential difference, as I said, comes because of the propagation of the electric pulse in the atrial wall and ventricular walls. The problem with relying on clinical electrocardiogram is that during diagnosis, the period is typically short. and the patient is resting, in which case the heart is not really exerting. So it is not the ideal place to observe the heart. If the person was freely moving and exerting, then we can determine whether the heart was responding properly. So scientists today use wireless electrocardiograms. They are developing wireless electrocardiogram, and my team is one of these people in this effort, so that we can attach wireless electrocardiograms and let people freely move and carry out their everyday activities. While they do so, we observe the electrical activities of the heart and see whether the heart is exerting properly. But the problem with this setting, the problem of using wireless electrocardiogram, is that when the people are free to move, then the electrodes placed on the surface of the earth, on the surface of the skin, vibrate. There is a slight involuntary vibration or motion, and this motion disturbs the measurement of the underlying cardiac activity and introduces its own erroneous signal, which we represent as motion artifacts. Depending on the intensity of the movement, their intensity also varies. So as you can see here, motion artifacts affect both the gradual baseline as well as the local waveform of the electrocardiogram. And sometimes we are not sure whether this modification or distortion comes from the heart itself or from the motion. The gradual change is relatively easy to remove. We can simply apply a high pass filter because the gradual move is a slow process. It contains low frequency components. So we can remove these low frequency components by applying some digital filters. But the local problems are hard to get rid of because their frequency component overlaps with the frequency of the electrocardiogram itself. So there has to be a mechanism, a more complex and sophisticated mechanism to remove motion artifacts. My team and I investigate different types of approaches. One of these approaches is dimensionality reduction techniques. Specifically, we use singular value decomposition and tensor decomposition. So the rest of this talk will be focused on these two approaches. To make the presentation tractable and to establish a shared understanding, I will begin with a simple observation. So what we do is the following. Motion artifacts are results of human motion. If the person were not moving, then we hardly observe motion artifacts. So if a person is moving and this movement affected the electrodes, then we can measure the movement itself using some movement sensors such as accelerometer sensors, gyroscopes, or skin conductance, because when the person moves, the muscle move, and this muscle change the electrical activity of the skin. So if we measure the impedance or the electrical resistance of the skin, then we can correlate it with the motion itself. So different types of motion sensors can be applied to measure the motion. So if we correlate the motion artifacts included in the ECG with the motion measurement we took, and we subtract the motion artifact accordingly, it is possible to get the clean ECG signal. This is our hypothesis. So what we do, we use the Shimmer platform here. You see, the Shimmer platform is produced, first developed at Harvard University. Now it's a company on its own. It produces different types of wireless medical platform, amongst which is the Shimmer 3 platform. It integrates the 3D accelerometer sensor, 3D magnetometer sensor, and 3D gyroscope, along with a 5-lead ECG. So that means we can measure motion, the localized motion in this area, in the upper torso area, along with the electrocardiogram, and then try to correlate them so that we can remove the motion artifacts affecting the ECG. So here, for example, these are the electrode we place at 3 position. So the measurement taken between these 2 electrode is called the left R, right R. The measurement taken between this electrode and this electrode is called the left leg, left arm. And the measurement taken between this electrode and this electrode is called the left leg, right arm. So we have 3 different channels of ECG. In addition, now we have the 3-dimensional accelerometer in the x-axis, the acceleration of the body in the x-axis, the acceleration of the body in the z-axis, and the acceleration of the body in the y-axis. Similarly, we have the gyroscope measuring the angular velocity of the body in the x-axis, in the y, and in the z-axis. And we try to establish correlation between this to reason about motion artifacts. We use also, the problem with this type of setup is that the heart also creates its own motion when it pumps. And this motion will be captured by the accelerometer and the gyroscope, as well as the impedance sensor. Now, it is difficult to distinguish between the motion affecting the electrode from outside as well as from inside. We are interested only in the outside because the outside is the one which affects the electrodes. The one inside is not responsible for the undesirable movement of the electrodes. So if we take motion from this point, however, at this point at the back, we don't sense strong electrocardiogram signals because the electrocardiogram signal does not travel that far. But we can measure the motion. So once we have the motion at this point, we can even mix it with a clean ECG signal so that we can test our approach using this technique. And then once if it works, then we can use the second approach for everyday use. Okay, so this is an example. Here, as you can see, this is a clean ECG signal we take when the person is resting. This is not affected by motion. We can generate a motion using this setup here. And then we have this sensor, this data from the x-axis of the accelerometer. Now we can deliberately mix these two to corrupt the ECG signal and then apply our approach on this one to see if it can separate them. If it separates them, then the approach works. Then from then onwards, we can use it for any type of movement and for any type of deployment. This is our purpose. Now, before we speak about our approach, I have to speak about dimensionality reduction techniques. Imagine we have this set of data. Okay, this constitutes a simple matrix. But this data may refer to some sensor data. Some sensor data, for example, if we consider these are the samples, we sample a sensor at every interval. For example, every 1 millisecond, every 1 microsecond. And these are different channels. Okay, for example, we have used electrocardiogram, electroencephalography, electromyogram, oximeter, and many other sensors to monitor a patient. And we synchronously sample these channels. C stands here for channels or device. Because we synchronously sample, each device emits a certain number at predefined time intervals. Now, because all these devices are observing one and the same patient, do they agree in their observation? Or do they deliver some complementary observations? So here you see now this matrix corresponds to some underlying reality. So the rows here indicate the channels, and the columns here indicate the samples. Now, one important way of seeing whether or not they are observing one and the same situation is to plot the data. For example, if we want to know whether the patient is well at this point in time, we can plot the observation of all of them. That means we take just the vertical data and plot them. Or if we want to focus on one channel for a long time, we can take all the data along this line and plot them. So now if we plot the vertical data, what we get is this one. At least we have, I have taken here 3 columns. And it so happens that the waveforms are the same. The sample values are different, as you can see, but the waveforms are the same. So what we were not able to see here by merely looking into the raw data, now we could see the data actually represent one and the same thing. At least the way the things change in time seem to be just the same. So instead of just looking at different columns, it would be sufficient just to look at one column. Similarly, we can also plot the channel values. And then if we do so, this is also what we get. The waveforms are the same, but the values are different. And yet the vertical waveforms and the horizontal waveforms are different. Now, indeed, if we closely look at this matrix and then factor out the numbers along the rows, here, if we just factor the same number, the number which is the same for all of them, we'll get 5. If we do here, we'll get 3, and so on. If we factor along the columns, this is what we get. So actually, the unique values of this matrix are the one depicted by the green with the green color. So we can get rid of the whole matrix, because in fact, the whole matrix is just the cross product of these two vectors. So here, 5 times 2 is 10, 5 times 1 is 5, 5 times 8 is 40, and so on. If you just take this row and this column, you'll get the value 9. 9 times 6 is 54, and so on. So the whole matrix here can be represented as a vector product or a cross product of two basis vectors. So we can remove it. We don't need it. We don't need to store all this data. Rather, it's sufficient to just store two basis vectors. So we can say that the matrix has just one feature only. And this feature is depicted here in green. And the green are called orthogonal. They form 90 degrees. You see here, ortho means 90. Gonal is angle. So this is a 90 orthogonal vectors. Orthogonal basis vector. If we have this basis vector, we can reproduce the original raw matrix. Not all matrices are so simple to decompose. Some of them could be like this one, more complex. It's difficult to factor anything. But with a careful separation, we can even decompose this matrix like this. So it consists of the addition of two matrices, and each matrix now you can factor out. Here, from here, 2 times 3 is 6, 2 times 7, 40, and so on. So instead of now storing these two matrices, you can just store these basis vectors. An interesting aspect of this basis vector, statistically speaking, is that now these two are orthogonal to each other. These two are orthogonal to each other. If we were going to normalize this data between -1 and 1, these two will be orthogonal to each other, and these two will be orthogonal to each other. That means there is no redundant information in this basis vector. And interestingly, these two basis vectors, the columns here, are statistically speaking independent from one another. This one doesn't contain anything belonging to this one, and this one doesn't contain anything belonging to this one. The same can be said of this and this one. So now more complex matrices can be decomposed into basis vectors. And once we have the basis vector, we can even plot and see whether they are similar. As you can see, they are not similar. So this is the first basis vector of the columns here. I'm plotting this column and this column. So you can see that they are not overlapping. And if you multiply and integrate them after proper normalization, you will get a 0 result. So that means we can search for hidden features, hidden independent features in a complex matrix coming from some sensors. If we have now multiple features here, for this case, you see here we have multiple features now, we can even plot them like this. So this is the first feature in the green. This is the horizontal, this is the vertical, and this is the horizontal. This is the vertical, and this is the horizontal. We can plot and analyze them. And in fact, we can also see how much each channel, we had 8th channel, how much each channel contribute to describe the first feature, how much each channel contributed to describe the second feature, how much each sample, these are the samples, each sample contribute to describe the first feature, and how much each sample contributes to describe the second feature. So by closely examining these features, it is possible to extract useful insight from raw data. The most important aspect of this is that we haven't made any assumption about the data. We just work it on the data which is available to us, which is why it is called unsupervised learning in machine learning language, because we are not conditioning the raw data by making some assumptions. We make absolutely no assumption whatsoever. One of the dimensionality reduction techniques, which is often applied, is the singular value decomposition, or in short, SVD. SVD, here you remember, we have now, this one is the vertical basis vector, this one the horizontal basis vector. So if there were many basis vectors, this would be a metric, a matrix in its own right, and this would also be a matrix in its own right. So SVD composes a raw matrix into many horizontal matrix, into a horizontal matrix and a vertical matrix. But now it has also another component, which is called the diagonal element, or the singular elements. The singular element here, it's a diagonal matrix. It assigns proper value to each basis vector. Not all basis vectors are equally significant or equally important. In some cases, we can reconstruct the original matrix by just using a couple of basis vectors. Remember, let me just take you back one. Remember, we could reconstruct this raw matrix just from this one set of basis vectors. But we said we need now two set of basis vectors to reconstruct, but not all of them, both of them are equally significant. For example, if the sigmas here, the sigma values here are different from one another, for example, if the difference between sigma 11 and sigma 22 is in the order of 1000, we can approximate the original matrix just using two sets of basis vectors and then reject all the other ones. So the usefulness of SVD is that it assigns singular values to the basis vectors. So now the original matrix can be described like this: sigma 11 times the cross products of U1 and V1 plus sigma II and the cross product of the UI column of the U matrix and the transpose of the VI column of the V matrix. How does this help us in dealing with motion artifacts? It helps us as follows. We have said our raw matrix here can be built by taking measurement from different channels. For our case, for example, these channels are the ECG lead from left leg, left arm, left leg, right arm, left arm, right arm, the A-axis of the accelerometer, the Y-axis of the accelerometer, the Z-axis of the accelerometer, and so on. And these are the samples. We sample the sensors at the regular interval. So we have this raw data. Now look, The 3 ECG channels here should observe one and the same thing, which is the ECG. The motion sensors here may observe both the ECG activity, the activity of the heart as it pumps, and the exertion, the movement of the body when the person moves. Now, with the use of SVD, we can decompose the raw data into highly correlated ECG signal and uncorrelated independent motion signal, which has nothing to do with the ECG. Because the ECG and the motion, they have nothing to do with each other. The motion is produced because I move. The ECG is produced as a result of the underlying electrical conduction system of the heart. Because SVD examines the existence of correlation along the horizontal and the vertical dimensions, we can take advantage of this correlation to separate the motion artifacts from the useful ECG signal. So this is, for example, how we decomposed. Here, this is the most important one. Here you can see that it is flipped, but you see that this is a proper ECG signal, okay, with QRS complex here, flipped, T wave around this area, and P wave is around this area. P wave usually disappears when the person moves, even in the absence of motion artifacts, so we can leave it. But otherwise, you can see that it's a clean signal. All the others we can consider as motion artifacts or noise from wherever they come from. We don't care. So SVD, by properly decomposing a raw metric, helps us to filter out the useful ECG signal without making any assumption. The problem with SVD, unfortunately, is that it's only two-dimensional. So here you can see it's just a two-dimensional. Suppose we want here, the raw data now is a time data, a time series. Suppose we wish to also take a look into the spectral aspect of the data. That means now we need an additional dimension. So for example, if we have this ECG signal you see here, we can apply wavelet transform or short time discrete transform to get some insight into the frequency distribution of the signal, so that we know not only what happens at a given time, but with what frequency or with what speed it has happened. So if we have this time series data, we can apply wavelet transform to get this two-dimensional data. So this one helps you the time, gives you the time aspect like this one, but this one now gives you the frequency aspect of the signal. So this is only for one channel. Now this is, for example, for left leg, left arm. If we want to have the same thing for left leg, right arm, then we have an additional two-dimensional data. For the accelerometer, we have an additional data. For the gyroscope, we have an additional data. That means we are now dealing here with three-dimensional data. So here you see that this is the time series. This is the spectrum. And now for each channel, we have to use another dimension. SVD does not help us with this one. But what does help us? Tensor decomposition help us. This is called a three-way tensor or a three-dimensional array. In the same way we decompose a two-dimensional matrix into constituting blocks, we can also decompose tensor. We can also decompose a tensor into constitutional blocks. One of these approach is called the canonical polyadic decomposition. Given this three-dimensional tensor, now we can express it as a multiplication or a cross product of three basis vectors. So this one is the vertical and the horizontal, like the case of matrix decomposition, but now we add a lateral component to it. Now here we have three different basis matrices. The matrix A relates this dimension with the hidden features. The matrix B relates this dimension with the hidden features, and the matrix C relates this dimension with the hidden features. Now remember, this dimension is a frequency dimension, this one is a temporal dimension, and this is a channel dimension. Now we are able to diagnose the ECG data from three different or orthogonal views. And this helps us to know the motion artifacts more cleanly and more clearly. So what happens is here we have now the corrupted ECG signal. We intentionally corrupt the ECG signal. And this is the motion sensor we used to corrupt this signal. Now the tensor decomposition cleanly separate them like this. This is the extracted signal from this one using tensor decomposition, and this is the motion signal which we use to corrupt this signal. So with tensor decomposition, it's possible to dissociate them. Likewise, here you can see now, this was the original clean signal, this was the corrupted signal, and now this is what we approximated. At least as regards the QRS complex, the wave complex which is associated with the ventricular activities, can be detected here cleanly. So at least we can use a wireless electrocardiogram for emergency purpose or for a telemedicine, so that doctors can observe a patient from a remote location and see if everything is all right, or whether the patient can be summoned to a clinic for a more closer examination. So with using dimensionality reduction techniques, it is possible to separate motion artifacts from the useful ECG without making any assumption, except the fact that we assume the motion artifacts are superimposed, or the corrupted signal is a superposition of the clean signal and the motion artifacts. This is the only assumption we make. It's linearly added to the clean signal. Other than that, we make no other assumption. Now, our next examination was to see which of the motion sensors were useful and for what type of movement. As I told you, we have considered accelerometer sensors, gyroscope, and impedance sensors, among others. And then you can see their capacity to reproduce the useful ECG signal measured by the signal to noise ratio. And we observe that it all depends on the type of movement. We consider different types of movement here. We consider jumping, running, bending, standing, stairs, climbing up and down stairs, and walking. They can be detected with different level of accuracy, but for bending, for example, we can use all type of motion sensors to remove motion artifacts, but the most important channel was the bioimpedance, followed by the gyroscope and then by accelerometer sensors. So in this research, we apply dimensionality reduction techniques to remove motion artifacts. We also consider different types of sensors, motion sensors, to correlate the motion artifacts with that of the motion of the person. And we consider different types of movements to examine the scope and usefulness of our approach. In conclusion, we allege or we assert that dimensionality reduction techniques can be useful to separate the ECG from the motion artifacts. SVDs are useful, but they work only in 2 dimensions. Tensor decomposition, on the other hand, are more robust and include additional information such as spectral components into the analysis, so that we can look at the problem from 3 different perspectives. By this, I come to the conclusion of my talk, and I thank you and say goodbye.